For stem cells, we need to know really about induced pluripotent stem cells. What does it do and what's the use of it? So a stem cell, we know from GCSE that these are cells that are unspecialized, and they can specialize into a range of cells. And also, number two, they have the ability to divide repeatedly. Very important feature. So stem cells have to divide repeatedly, and they can differentiate into specialized cells. Is a two defining feature. We need to know these four types of stem cell, and a really key focus is going to be on the pluripotent. The totipotent stem cells, these are embryonic stem cells, and basically you get them from the embryo, and they can differentiate into any type of cell. But also a unique thing about them is they can differentiate into extra embryonic cell. What does that mean? That literally means the placenta. They can also make the placenta. Next one, pluripotent stem cells. This is the one that we need to focus on. It's also in the embryo. So how is that different from totipotent? So pluripotent, they can divide into any type of cells in the body, with the exception of placenta. They cannot make placenta. That's the only difference about them. So they can make into a heart cell, liver cell, a kidney cell, a skin cell, a hair cell, any cells, but they cannot make placenta. So scientists really like these cells, so they can be used to treat human disorders. Multipotent adult stem cells, we know that adult stem cells can be found in the bone marrow, and this is showing you here an adult stem cell. They have limitation. They can be differentiated into limited range of blood cells, like B cell, T cells, white blood cell, red blood cell, neutrophil, basophil, and so on, platelets. They can't make like a liver cell, a heart cell, a skin cell. They can't do any of that. So it's limited to a blood cells that are found within your blood system, within your bloodstream. Last one is unipotent stem cell. These cells can only become one type of cell. Let's say your skin is replacing, so you have a stem cell in the skin, and that stem cell can differentiate into which cell? Skin cell only, not heart cell, not liver cell. Cardiomyocyte is a stem cell of the heart. When the heart cells die, these cardiomyocyte can differentiate into new heart cell. That's called unipotent stem cell. So the key idea is this, or he found the underlying principle is this. If in this cell, let's say it's a pancreas cell. Now pancreas cell has how many chromosomes? 46 chromosomes. It's got all the genes. Every single cell in the body, if it's body cell, somatic means body cell, it's got 46 chromosomes. So why is it the pancreas only makes certain things? It's because all the other genes are switched off. Why they're switched off? Is because they don't have the appropriate transcriptional factor. Scientists use the idea and they say, okay, we can change things around now. Let me show you this one here. Like pluripotent stem cell, remember it can become any cell except the placenta. Why is it? Because all the genes are activated. Why are they activated? Because they have all the appropriate transcriptional factor. So now scientists thought that, okay, we like these cells, pluripotent stem cell, but its problem, why is it a problem? It's unethical, and it's also, having a embryonic stem cell and being planted into a different person can cause immune rejection. Problem like that. So to overcome that, they goes, okay, why not we get a adult stem cell? Why not we get a unipotent adult body cells? And then we can, obviously this cell is unipotent because it can only make how many cells? One cell. And then we can add in different transcriptional factor. All those genes that are switched off, we can switch them on by adding different transcriptional factor. So in other words, we can switch on any gene that we want. Any gene, let's say the patient has a spinal injury and we want to regrow the spine. Okay, we get the unipotent stem cells and we add a transcriptional factor that will stimulate, that will express the gene expression of that cell of the spinal injury part. And now this cell can become any cell. So in other words, iPS is now like a embryonic stem cell. Wonderful thing to do because less ethical issues, no embryos are used, and less chances of immune rejection.